Hello. Let's talk about our next topic, 1b.3, experimental and correlational studies. Today we're going to focus on just that first one. This is 1b.3a. We're just going to talk about experimental studies, primarily research, primarily the experiment within research. We're not going to focus on correlational studies quite yet. So recall so far we've talked about descriptive types of studies. They describe things like the survey, naturalistic observation, and case studies. So today we're going to focus on the biggie, which is experiments. Okay, these are probably the things we're going to study the most in psychology because they can show, and remember, I've told you this multiple times, underline and circle it, make sure you can remember it. They can show cause and effect. Okay, they can show cause and effect. They do this by manipulating, controlling variables. So, experimental research. The big idea here is we are trying to prove cause and effect. We are trying to prove that one thing causes another thing, or that when one thing is happening, this is going to cause another thing to happen. Whatever this first thing is, it will shift our thoughts or our behaviors enough to shift those. Okay, and we have to prove that by controlling as much as we can. Okay, manipulating a certain variable in order to and controlling everything else in order to impact something else. We're going to get more specific with that, but bear with me. So, big idea. Manipulate an independent variable to see how that impacts what we're studying, the dependent variable. And we do also control as many variables as we can. This means that, like, when we're studying things, stuff will always get in the way, right? Like what people had for breakfast that day, what they've been watching on the news, the environment that it's happening in. You can't control everything, but the more things that you control so that all the people in your study are going to like have less things impacting them. So we can say that almost with certainty, what we manipulated showed that this independent variable, this variable, impacts the other one. Okay. One causes a change in the other. That's the goal of an experiment. We recall operational definitions, how important they are, been on the test for the last 7 years. We have to define both an independent and a dependent variable. Okay, so what these are crucially important. You're going to get plenty of time to practice these, but let's just go over quickly what they are. The IV and the DV, independent variable, is the if-then. Okay, so the independent variable is first, and that is what we are manipulating. Okay, so we are manipulating an independent variable in order to figure out if that changes the dependent variable. Okay, so if we shift something in the independent variable, then your dependent variable will shift. So on a purely like medical or scientific thing, if we're doing this as simply as possible, if you work out in the mornings, say you get up and do a morning workout, right? You go for a 3-mile run every morning. We're setting up a study to figure out if my hypothesis would be that eating a bowl of oatmeal 30 minutes before you run will increase your time. Okay, will make you run more effectively. We'll say like improve your ability to run without effort. I don't know, like something like that. So the independent variable would be that eating the oatmeal. Okay, I'm figuring out if you eating that specific breakfast, I'm operationally defining it as a bowl of oats made with almond milk, and maybe chocolate chips, right? Like nothing else. Everyone in my study, some of them would have that manipulated, and some I would manipulate it and not give them the breakfast, and see if when they all went for a run, if it changed their times. That would be their dependent variable, their ability to run. So dependent variable is what you're studying. Independent is what you're manipulating. Dependent is what you're studying. So again, these are extremely important. You got to understand what they are. The question is, how does the if impact the then, the x and the y? How does a change in one variable affect another variable? So just to pull it back and to give another example and think about this a little bit differently, I can say oftentimes these are referred to as the x and the y, x being the independent variable. This is what is manipulated in order to see its impact on the y. Okay. So manipulating the x or the independent variable impacts the y or the dependent variable. If we change an independent, then it will have an impact on the dependent. You don't do anything to the dependent variable. That's what we're studying. That's what we're watching to see if it changes naturally. But you are manipulating. You're changing in some way an independent variable. So independent variable is what you are manipulating. The dependent variable is what is going to be. It's dependent on the independent. Okay, that's why it's called that. The dependent variable is going to change based on what you manipulate in the independent variable. So we are measuring or researching that dependent variable. So other things you should know in there, we talked a little bit about random selection or sampling in surveys. We've got to randomly pick out who's going to be in the study. And I told you, don't get that confused with random assignment when we get there. So random assignment, this is how we're selecting what people are going to be, what group they're going to be a part of. So in a good experiment, you have to, again, we're controlling as much as we can. We can't just give a bunch of people oatmeal and study them. We have to control as much as we can and also show a group that doesn't receive the treatment, right? This has been a huge issue in studying things around COVID, right? It's like everyone wants to get whatever the hot new treatment is. People aren't going to agree to be a part of a study and not get whatever that new treatment might be if they have COVID, right? So we haven't been able to set up these really good studies where we have control and experiment groups in order to determine what is effective, right? What independent variable or medication causes, lower mortality rates in COVID, the dependent variable. So the groups that we're always going to have are going to be a control group, a group that doesn't receive whatever the treatment is. That means their independent variable is not manipulated, okay? It's not changed. We're not giving them whatever the thing we're testing is, right? They are just getting nothing, right? In some cases, if we're giving them medication, right, we would just give them a pill with sugar inside or just nothing, right? Like they need to think they're getting something, but they can't know that they're in the control group, but they still need to be a part of the study so we can see what happens. We'll talk about placebo here in a minute. Experimental group is the one that receives the treatment, right? So back to our example of the oatmeal and the runners, our control group in this study, if I was studying this with 50 runners on a team, I could take 25 of them, and I would of those, I'd say I'm going to test all of them. I'm going to test the whole team. And then of those 50, I would randomly assign. I don't get to hand pick which one goes in one group. I would just turn them into a number, and I would say the 1 through 25 are in the control group, 26 through 50 are in the experimental group. 1 through 25 in the control group, they wouldn't get oatmeal. Okay, they would just they wouldn't do anything. They would get no food whatsoever. Experimental group would say you must eat the oatmeal, right? So control group doesn't receive treatment, experimental group does. And then we're comparing what the differences between these two are, right? So control group, no treatment, experimental group receives the treatment, and then we study what the outcomes are to see if the control group and the experimental group had significant differences. Okay, that's how we're determining the results. And the ultimate goal that we're trying to do is we're trying to figure out if our tests are valid. Okay, valid. We are searching for validity. Validity, or a test being valid, means that it's testing what it's supposed to test. There are a lot of tests that can get interesting results, but if they're not getting what they're supposed to be, then they're not effective, right? Then it means that we have to get rid of all of these other biases and confounding variables that get in the way. Otherwise, it may not be valid. So valid is that it tests what it's supposed to test. Okay, so it actually shows what your hypothesis is claiming and not just some other random finding. So that's validity, right? We've talked a little bit about reliability, meaning that you can test it again and again and again and get the same results. Validity is that it's testing what it's supposed to. Okay, so couple examples just so you can think about this, right? Here's one. A researcher wants to prove that violent films increase anxiety and heart rate compared to non-violent films. Okay, researcher wants to prove that violent films increase anxiety and heart rate compared to non-violent films. So that's what they are studying. Their hypothesis would be that violent films increase anxiety and heart rates. Okay. In their study, they're going to set it up so that one group watches the violent Saw whatever we're on now, right? Like Saw 33. While the other group is going to watch, and a non-violent, it's American classic, The B Movie. Okay, think about it. Which one is the control group and which is the experimental group? Okay, take a minute if you need it. Which one is controlling? If our hypothesis is that violent films increase anxiety and heart rate, okay, violent films increase anxiety and heart rate. What's our control group and what's our experimental group? Okay, what's our independent variable? What are we manipulating? Which group would be getting the test and which one would not? Think about it. Okay. Saw, whatever number we're on, is our experimental group. That's the manipulated variable. We are manipulating this type of anxiety. We're manipulating the violent film. That's what we're adding in. That's the test. That's the treatment. We want to see that if non-violent films will lead to increased anxiety, if violent films lead to increased anxiety. So if you get the violent film, Saw, that you're the experimental group. However, if you sat there in this lab, whatever you were part of a study, and you watched the B movie, you were in the control group, right? You just sat there and watched a movie, because we have to see if maybe it's some of the other factors. Maybe it's the fact that you're being studied, that you came into a lab and sat down, and maybe that's what's causing everyone to be anxious, right? If after we watch both of those movies, everyone showed an increase in anxiety, right? It's probably not the B movie that's causing that. It means that there's some other variable that's causing it. But if we show big spikes in the experimental group that watched Saw and anxiety and heart rate, but not in the B movie, right? We've got we've controlled as many variables as we can. Then you can make a claim that when watching extremely violent movies, heart rate and anxiety increase, right? We can show cause and effect. And then hopefully people are able to replicate that study, and they would say that is a reliable finding. Other examples. Here's some sampling assignment type examples. So if we're trying to figure out headphones and the ability to, if those can help you or harm you to study in some way, right? If you're setting up an experiment, we take all students, okay? We're going to take all the study hall students at Chapel Hill High School. You would first have random selection, okay? So random selection and sampling, okay, is different than random assignment. You've got, out of all study hall students, we don't have, we're not going to have 700 pairs of headphones we can give out. We have 40 pairs of headphones to do this study with. So we would randomly select 40 of them, draw them out of a hat, okay? So we've got the full population, all study hall students. 40 students randomly selected. That's our random selection. From that, you then do random assignment. You assign them into either the experimental or the control group. Okay, so half of the students are randomly assigned to the experimental group. Half of them are assigned to the control group. Remember, the experimental group is going to get the treatment. In this case, the prediction is that the headphones are going to make it harder for you to study or focus during study hall, or that it will harm your grades in some way. So those 20 students were given headphones, and they were told to wear those headphones and listen to music every day in study hall. Okay, so those 20 students got headphones. They listened to headphones every day in study hall. And then we had to see what their average grades were at the end of the quarter. And then we did the same thing for the control group. We took, again, 20 randomly out of those 40, which were a part of the full population. Of those 20, we said, you cannot wear headphones in study hall. And at the end, we're going to look at average grades at the end of the quarter. Okay, so we've got random selection out of the full population, then they're randomly assigned into either the experiment or the control group. Experiment gets the treatment. In this case, it's headphones, the independent variable, to see if the dependent variable, the grades at the end of the quarter, are impacted, right? If those 40 students all had, if their GPAs or their grades were pretty much the same, then it wouldn't really matter, right? They're clearly showing it's not that big of a deal. But plug this in. But if we showed that a lot of the students, that on average students that wore headphones did better or worse than the students that were not allowed to wear headphones, we can start to see a finding because we controlled for a lot of other variables. We set this up randomly selected, randomly assigned. Okay. In setting up these experiments, one of the big things we have to do is we are trying to reduce bias. Okay, that's true in most of these settings, most of these types of research. The biggies that we're going to do: one, random assignment. We've mentioned that. That is probably the most important thing you can do. It's that whoever you've selected to be a part of your study, you're just randomly placing them in groups. You don't get to pick, because no matter what, no matter how unbiased you try to be, you're ultimately going to want your study to come true. The findings, your hypothesis, to be correct. So by doing this, you remove any ability for you to make an impact there. This also eliminates a lot of confounding variables, and these are something that we'll talk about these a little bit more, but confounding variables are anything that can get in the way. Okay, these are things that anything that would complicate your ability to study that. So to go back to like my oatmeal example, confounding variable could be like whether or not a student was somebody that ate breakfast at all, right? Whether or not a student has always been eating oatmeal or never ate breakfast, things like that. So we've got to figure out what other variables might impact how they took this survey, how they participated in this experiment. And then other ways to reduce bias, or things we need to recognize and think about it: this Hawthorne effect or the observer effect. Oftentimes people, when they're being observed, change their behavior no matter what, right? We shift how we observe, knowing that someone is observing us. Famously, what really showed this was there was a production facility, and they wanted to study to see what type of light made people work more effectively. And they had people, like researchers there, like taking notes consistently over the course of a couple weeks, and they found that almost no light at all improved people's ability to their production. They also found that moderate light, high light, and like blue light all increased production. They were like, this can't be right. Right, this is weird. We figured having more light would make production, but what they found was that people just knew that they were being watched. That's what produced production. It wasn't the light. So being observed is a big part of what can change the way people act. Think about your teachers. What happens when the principal walks in the room? Teacher goes from like sitting there like, yeah, what's the next answer, to like, please, little Johnny, like enlighten us with how it made. We like put on this big face and show, and we can't help it, right? Like, I like to think that I'm a good teacher most of the time. But when the principal walks in the room, you can't help but like want to show off how good of a teacher you are. And everyone does this in just about every effect. You all do this too, right? Even like, it doesn't matter what the principal thinks of you all as a student, right? But I see you all sit up straight and try to answer questions when you had never done that before. And we're being observed. No matter what, it changes how we do things. So there's nothing we can do about that, but you have to be aware of it in experiments. Another big you need to watch out for is placebo effects. If people are if they get any treatment, oftentimes if we know we're a part of a study, we're going to think it's going to do something. For instance, placebos have been shown to have a huge impact, a significant impact, like a pretty significant impact on depression, right? They're not as effective clearly as like serotonin reuptake inhibitors, but they are pretty effective for a large part of the population, right? For a lot of people, just taking a pill that they are told will make a difference in depression will improve their depression, okay? Right? Someone just telling you this pill is going to make you feel better. And it's not anything. It's just a sugar pill, and it will improve it. Just the mental processes. Again, how we think about things. That's cognitive psychology. How we process information really changes things. It changes our like stress levels. That's why meditation is so important. So one of the big things that we have to watch out for is this placebo effect. People thinking and looking out for impacts, right? So when they didn't really get an effect, still being impacted by the fact that they know they're in a study and they're receiving a pill and maybe thinking it's going to do something. So you got to account for that and think about it. The color can do things. Red pills showed more of a placebo effect than like a white or green type of pill. So got to think about that. Yeah, capsules versus tablets, like all of these things can come into effect, and people thinking that they got something when they really didn't. Reducing bias. Finally, single and double blind studies are probably, these are like the gold standards, right? Double blind study is the most effective, the most unbiased way to do anything. I would recognize the difference between these single blind studies are where participants don't know if they're in the control or experimental group, but the researcher does. Okay. So a placebo here could be like they think they're getting treatment can reduce symptoms. Yeah. So a double blind study is where both participants and researchers don't know who's in what group. right, until the end. Everyone's just put a number, and then you get your findings, and then you kind of like open the envelope to see who was in what group and what their findings were. And this really shows that the researcher could have no biases in there because they couldn't possibly manipulate it because they don't know who was in what group. Okay, so double blind is the gold standard. Single blind is still very good. And again, in COVID research, this has been a big problem because we haven't been able to perform a lot of these, or really even single blind studies. It's hard for us to do a lot of research on things like hydrochloroquine, because when you tell someone, like, would you like to be a study as they're lying in the ICU, struggling to survive, they're like, just give me any treatment. I don't want to be in a control group. I want to know that I'm getting something. So it's tough. So big ideas. The goal of experiments is to show cause and effect relationships. We set up an independent variable that we manipulate in order to show the dependent variable. Okay. And then we are trying to remove those confounding variables, and we are ultimately trying to show cause and effect. To do that, we have to remove our biases. We do that by thinking about and recognizing placebos. We set up blind and preferably double-blind studies and recognize the Hawthorne effect. We use random selection to pick who's going to be in it. And then more importantly, we randomly assign into groups. Got to know the difference between those, selection and assignment. We then randomly assign into groups, and we have to have that control and experimental group, right? We've got to prove that there is a difference between those in the dependent variable, which is what we are studying. So we're going to call it there. I know this is probably a long one. This is an important topic. We're going to use this stuff forever. You've got to know the experiment. So this is a video you may have to go back, rewatch, be prepared for that. But if you got questions, bring them to class. You will do some practice on this stuff. It is important. We got to know it. With that, I will say farewell. Thank you all for your eyes, and I will see you in class.